intimate friends and family defense of the history of the joint program, but I'm so pleasantly surprised. This is wonderful. And I'm so glad you're here to share this big day with both of us. Emily first came to my lab as a summer student fellow in 2009. At that time, she was a junior at Hood College, but she already had a deep interest in these little organisms that she's going to talk to you about today. And when she came back a year and a half later to enter the joint program and started to talk about what her thesis might encompass, it became clear that deep interest was not a passing phase. And I'm happy to tell you that she stands before us today, I think the world expert in Ciliate of the New England shelf. So maybe that sounds super specific to you. I wanted to just say that Emily is also a very broad-minded and adventurous scientist. And one way that she's shown us that is that while she was in the joint program, she's co-author on 3 papers that do not appear in her thesis. Some of you will remember several years ago, a group of students stood up here and gave a public debate about the influence of spare drip on phytoplankton ecology, and Emily was one of the students who followed through on that fascinating debate to bring it to publication. She also took the opportunity to go to the Arctic with Sam and studied under ice phytoplankton blooms and published with a group of colleagues on that. Truth be told, she went to the Arctic looking for ciliates, but when the phytoplankton were more interesting, she dove right in and produced on that. Today, of course, Emily's going to talk about her thesis research, not those other papers. I think when you listen to her talk, you'll see some elements of really classic natural history running through Emily's research, but at the same time, she's injected incredibly modern approaches that completely revolutionize the kinds of quantitative insights that you can get from a natural history approach. And I'm sure you're going to enjoy hearing about that from Emily.
Thank you, Heidi, and thank you, Sam. I'm so amazed that everyone is here today and so excited to let you know about my research. And so we're just going to jump right into it. Oh, well, we'll jump right into it in just a second. There we go. Wait. So, yes, okay. So why do we study ciliates? Well, the classic food chain will show a transfer of carbon from larger autotrophs to large zooplankton. And in the 1980s, this classic view of the food web was challenged with the discovery of the microbial loop, in which ciliates are top predators. Ciliates create a vital link between smaller autotrophs and higher order predators who cannot consume these smaller cells due to morphological differences and efficiency. They're also very important in nutrient regeneration. They help to fuel primary productivity in nutrient-limited waters. Ciliates are part of the larger microzooplankton community, and microzooplankton have been known to be large consumers of primary production, at times consuming up to 50 to 100% of daily primary production. And there are suggestions that interruptions in grazing pressure can initiate the North Atlantic spring bloom, and in turn, when grazers are coupled with their prey, they are powerful enough to suppress it. Ciliates are also highly diverse. They're one of the most diverse protists in aquatic ecosystems, and this provides a valuable target for studying community dynamics and ecosystem functioning. And so while this is just a very simple schematic here, you'll see in my talk how diverse they are. So at the beginning of my research, I had a few driving questions, and they were what types of ciliates are the major players in aquatic ecosystems, what factors regulate their community structure over time, and do certain taxa display distinct patterns. Many times, we don't even know the basic community structure and how this holds up over time. So my work tried to quantify and describe these patterns. And I study ciliates of the New England shelf, which shows high seasonal variability of biological processes and environmental conditions. here is just an annual cycle of temperature, which you can see in this winter. Sometimes we'll even reach to about 0 degrees Celsius, and in the summer sometimes we'll have higher than 20. We also see variation in the phytoplankton community here. And this figure is just a proportion of the size class of phytoplankton contribution to the overall phytoplankton. And in the red line here, you can see in the winter is most abundant, and these are the microplankton, the diatoms. And if the diatoms decrease, we see an increase in proportion of the nanoplankton with the green line. We also have a very notable peak of proportion of picoplankton during the summer. And so since these coastal temperate environments exhibit such large variations in the environmental processes and primary production, it provides a valuable stage for studying the large signals of responses from ciliate micrograzers. And my study site is here, south of Martha's Vineyard. My observations come from an offshore flux tower, which is about 3 kilometers south of Martha's Vineyard in a 16 meter depth water column. And the Martha's Vineyard Coastal Observatory hosts a wide variety of meteorological, hydrographic, and biological instrumentation that can provide us real-time information about temperature, salinity, daily solar radiation, wind speed, wave height, and many more. And as I go through my talk, I will refer to the Martha's Vineyard Coastal Observatory as MVCO. But ciliates are traditionally difficult to study and identify. Net collection can be harmful to their structure. Some ciliates can slip through mesh about 20% of their size. They're also difficult to culture due to the inabilities to supplement their complex needs of nutrients and prey. Traditional Lugol staining and preservation can harm the cell through shrinkage, distortion, and even disintegration. Also, long-term handling can disrupt the cell. And when it comes to identifying some of these ciliate stem species, you need to be able to identify certain fine-scale characteristics that sometimes can only be seen through scanning electron microscopy and specialized staining. And with recent DNA sequencing technology, we also see that there can be cryptic species, where 2 species can be genetically distinct but morphologically indistinguishable. And so, what's really exciting about my research is that I can now study ciliates without some of these traditional limitations. I can study them in situ and in long-term high temporal resolution. And while it was originally designed for phytoplankton, we have deployments of the Autonomous Submersible Imaging Inflow Cytometer, IFCB. And again, as Heidi said, this revolutionizes the way we study ciliates because we can now observe them without the need to culture or preserve. And the way this instrument works, the fluidics are similar to a conventional flow cytometer. A 5 ml sample is injected into the center of particle-free sheath flow, where it is then focused and the cells go single file through a red diode laser. Then this laser then causes light to scatter, and chlorophyll-containing cells emit red fluorescence, which is then used to trigger the flash lamp, and a portion of the wavelength, the green wavelength, is then used for the camera. So essentially, as cells pass through the imaging flow cytobot, laser induces fluorescence in cellular pigments, and we can capture an image of the cell. And so now we have this new way of studying ciliates, and what's really interesting is we can take many pictures rapidly underwater. So I'd like to introduce you to some of the ciliates that we see at MVCO. And ciliates are a big box of diversity. And as you can see, IFCB provides excellent quality and high diversity of images. And this information is not only for taxonomy, but also quantitative in the forms of hundreds to thousands of images every hour for a decade. And nowhere else can we get this kind of information. And as I said, they are diverse in morphology, size, and shape. So when I categorized these ciliates, I tried to lump them in groups of similar morphology. But there are times where we see Things like this ciliate here, Laboya strobila, which has a distinct enough morphology that I can identify down to species. And so along with this taxonomic and abundance information, we also can get information about size of the cell. We can estimate biovolume and in turn convert that to biomass. We can also capture some grazing in action. And you can see this ciliate here. You can see some of its prey and the food vacuoles. And these other ciliates here are feeding on diatoms. But because ciliates are so difficult to study, long-term, high-resolution data sets can be rare. And this can limit our ability to understand ciliate dynamics and abundance over time. Typically, for ciliate time series, we either have duration or resolution. And limited sampling resolution can cause challenges because ciliates have been known to fluctuate so rapidly in the system. As you can see here, this is just a historical study over a year. At the beginning of the study, the researchers were collecting samples about every 2 weeks, which later on in their study lapsed to about every month or 2 months. And they were only able to capture these fluctuations when they had high sampling resolution. And ciliates are known to fluctuate rapidly like this because they can quickly respond to favorable environmental conditions, as well as quickly couple with their prey. But at MVCO, I've been able to resolve this because deployments of the imaging flow cytobot are continuous, multi-month unattended deployments, and back-to-back with limited sampling interruption. So now we have a time series from mid-2006 to 2016. And time series can give us a lot of insights into ciliate dynamics. Observations of co-occurrence patterns between predator and prey and repeated responses to environmental changes can give us insight into their ecology. We can also see if certain events within a time series are ephemeral phenomena versus whether they recur in the system. And ultimately, they can provide insight into long-term trends associated with environmental and climate change. And I particularly like this quote, ecological time series are the Cinderellas of ocean science, long neglected drudges eking out their existence at the edge of what is in fashion. They now find themselves in favor at the climate change ball. And this speaks to me because I always wanted to be a Disney princess, and now I'm studying these time series. And so to understand and enhance our time series, I focused on 3 lines of methods to help understand more about feeding preferences, seasonality, and how well morphotypes and genotypes correspond over time. And so in my second chapter, I described an ICB that we developed with automated staining capabilities, which allows us to observe live ciliates whether or not they contain chlorophyll fluorescence. And then I applied this instrument in various laboratory and field settings. I gave a departmental seminar about this a little while back, so I won't be talking about this chapter today. In my third chapter, I applied a statistical model to the 10-year time series of herbivorous ciliates at MVCO. And so I could understand more about seasonality and multi-year trends. In my fourth chapter, I explored seasonality from taxonomic information from high-throughput sequencing and compared patterns between morphotypes and genotypes over 3 years. And so, how do you make sense of a time series? As you can see here, we have 10 years worth of inter and intra-annual variability, which can provide a plethora of information. And I was more interested in how to quantify the regular or the annual cycle. And so, to understand more about these seasonal patterns, I needed to be able to separate the expected seasonal cell concentrations from interannual variability. And so I worked with Andy Solo and Kristen Hunter-Severa to incorporate a model that explicitly separates annual and seasonal effects. And this can be helpful in times when, over a time series, certain years are high in abundance versus the next might be low or non-existent. And so here's just a mock time series where you can see a fairly regular annual cycle, but is increasing over the time series. And to parameterize these cell concentrations over time, they were a function of this average cell concentration, which is our constant in this equation, and two different components. An annual component, which gives us information about the interannual variability, and the expected seasonal cell concentrations. And to fit our time series data to this model, we use the maximum likelihood approach. And then we can separate this model into the two components that I just briefly mentioned. Over here on the right are these annual multipliers, and they give us information about how different years vary in magnitude from one to the next. And as you can see, it well describes how this time series here increases over time. And then we have the expected seasonal cell concentration, which is the annual pattern that we see without this variability. And again, this is from data fitted by the model. And it can kind of give us information. So let's say you were looking throughout a time series and you picked a year. We most likely would have this seasonal pattern. And also in partitioning these two parts, this annual and seasonal component, we can then calculate confidence intervals that better represent this data. And if the if the data is over-dispersed according to the Poisson distribution, then we inflate the confidence intervals. And so I'll be showing these plots throughout the talk, and if anyone has any questions about what they represent, please let me know now. It's okay. So not only can I apply this model to count data, I could apply it to all 25 different groups that we see at MBCO. But I focus my question on three main groups. And first, I want to go back to something I didn't fully describe to you before. When imaging flow cytobot detects ciliates, because it detects images of cells that have chlorophyll above a trigger threshold, it only detects ciliates that are mixotrophic or herbivorous. So I wanted you to keep this in mind, that images from IFCB are from herbivorous ciliates. And so for the three groups that I looked at for these studies, I focused on three different functional groups that we see at MBCO. And so my first one is an obligate mixotroph, Lavoia strobila. And this mixotroph has been known to obtain about 20% of its growth through photosynthesis, and this fraction grows even higher when you take into account its entire carbon budget. And historically, in waters around Woods Hole, it's been found to contribute to greater than 50% of the biomass of chlorophyll-containing ciliates. It also, in our time series, is consistently in the top 5 contributors to biomass yearly. It also, because of its distinct morphology, works very well with our automated classification of images. So automated classification extracts image features, and with a machine learning algorithm, it can then place the images into their proper taxonomic categories. So for further analyses, I use this automated classification, and I do so at 2-week intervals for the model. And here's a time series of Lavoia. And in the black line is daily resolved counts. And again, nowhere else can you get this kind of data. And in the red stars here are our manually classified images, where we went in and manually verified that these were Lavoia. And they're at about every 2-week intervals. And so I have two major questions going forward. How do morphotypes contribute to seasonal patterns? And do these patterns vary from year to year? And so for Lavoia strobila here on the left is the expected seasonal cell concentration. I saw that there was a very strong spring peak that recurred every year, and though varied in amplitude. And then we also had a smaller varying in timing event in the fall. And when you look at the annual multipliers, we do see these multi-year trends. But what is most notable is that over the time series, we see a decrease in the Loboya abundance over the last 7 years. And this will be interesting to study further whether this is due to warming temperatures or possibly when we apply this model to certain prey types, we might see these kinds of trends. And so for my second group of ciliates at MVCO were the tintinids. And tintinids are loricated ciliates, so they live essentially in a shell-like housing, and they are strictly heterotrophic. For automated classification, tintinids as a total community work very well. But when I do identify them further morphologically, I use manual identification. And because of the resolution of the images and sometimes the orientation of the cell, many times I identify them down to genus, and sometimes within that genus I separate them by size. Again, here's a time series of tintinids, and I was asking similar questions from Loboya. And when you look at the total tintinid annual cycle, you see very distinct and narrow spring and fall peaks in abundance, which are fairly even. And also a smaller winter peak that, while not shown here, usually only occurs in certain winters. But what's interesting is when you look at these annual multipliers, and even though it exhibits similar seasonality as Loboya, spring and fall, we see that over the time series, their abundance is not changing very, very much. And then we also see variations in the seasonal cycle of total tintinids manifest itself when we look at different taxa. And so for the genus Tintinopsis is the most abundant tintinid group. And for this, I do split by size. And the largest size class, greater than 60, predominates this spring peak. And when you go to look at the medium-sized class, we see that it also contributes to the spring, but there's also a smaller fall peak. And then the smallest size class of Tintinopsis, less than 40 micron, seem to be occupying these warmer temperature niches in the later half of the year and have broader peaks of seasonality. The next genus that I look at is Stenosemella, and one of these tintinids I can identify down to species. So Stenosemella shows slightly different patterns than Tintinopsis, where we see that it occurs during the late fall through the winter into the spring. And then the large morphotype of Stenosemella also varies from the other species, where it's just occurring during these fall times. We also have some more rare tintinids, in which it's a little bit more difficult to capture their seasonality because of such high variability. But we have two tintinids that are hyaline loricated tintinids, so they have a clear lorica. And Mutintinus occurs during the late summer, early fall. Favella, we see it occurs from the spring into the fall. And Tintinidium musicola has a very small fall peak. And because I see such distinct seasonal variations between taxa, I wondered if this could then be continued on the cellular level, because sometimes imaging does have its limits. And so a new approach to, or modern approach to studying ciliates in the environment is genotyping. And I wanted to compare these two new approaches in the ways that change the way that we study ciliates. So for my third major question, I wanted to know how well do morphotypes and genotypes correspond. And morphotypes are designated by the images, and genotypes will be designated by the DNA sequencing. And tintinids provide a unique opportunity to do this, as they have very distinct morphological characteristics. And these kinds of studies have been done, though they're very rare, and they've never been done over such an extended multi-year time series. And And so to do this, I used Illumina high-throughput sequencing, which is a massive parallel sequencing technology. And I took 27 discrete samples from the tower, about every 1 to 2 months over 2.5 years. I then targeted a part of the small subunit ribosomal DNA that's specific enough to amplify DNA from ciliates of the class Spirotrichia, which in particular I was interested in the oligotrix and the choriotrix. And I've already described one oligotric to you, that's Lavoia, and tintinids are choriotrix. And as I said earlier, ciliates are very diverse, and I was only targeting this small section. But coincidentally, they predominate the herbivorous ciliates at MVCO, and they also typically dominate planktonic ciliates in general. So then these samples were sent out to the University of Rhode Island for sequencing, and when they came back, I processed them with computer program Chime. They went through a few steps of quality controlling and removing chimeras, singletons, non-ciliate OTUs. I then normalized the data with cumulative sum scaling, which allows us to compare different samples with different sequencing depths. And then these counts were 4'3" transformed. And first I'll show you 2 tintinids that we are able with the images to identify down to species. On the left axis here are the cell concentrations from imaging flow cytobot. And while it's not up here, the units are cells per liter, which are 4'3" transformed. And this is to keep consistency between the comparisons, as well as 4'3" transformation helps us to retain 0 counts. And so you can see for Stenosamella, we have that late fall through the spring seasonality. For Tintinidium musicola with these samples, we only captured the ciliate once during the time series. And when I compared these counts with the normalized OTU sequences, I saw very significant correlations between the 2 methods. And that for Stenosamella pacifica, you can see that the sequence counts are capturing the seasonality that images are. And for Tintinidium musicola, while it did capture this later half of the year peak for this ciliate, we also saw it captured it at other times of the year, which isn't completely surprising. The sampling effort between these 2 methods are different. For high-throughput sequencing, I collected discrete bucket samples, and when I looked at tintinids, I examined a whole day of tintinids, which can represent different parcels of water passing through the site. But this gives some indication that possibly there's a quantitative aspect to this high-throughput sequencing. And so I wanted to extend this further and see if there are relationships with genotypes from the different size classes. As I said, Tintinopsis, which is the most abundant tintinid, is only separated by size. So I looked at tintinids species that came out from the high-throughput sequencing, and I attributed them to the different Tintinopsis size classes that we see at MVCO. And there was only one species that associated with the smallest size class of tintinids, and this was Tintinopsis parvula. And then there was one ciliate, Tintinopsis cylindrica, that associated with the largest size class. And when I overlaid or when I compared with the normalized sequence counts, I felt there were significant correlations. And this might represent the fact that these 2 tintinids are dominating these size classes, or that they appropriately represent the dynamics of these size classes. And then there were 4 different species, which are represented by each of these panels, that came from high-throughput sequencing that I attributed to the medium size class. And when I compared with the medium size class counts, unfortunately, I didn't find any significant relationships. And I even tried to sum all of the OTU counts from the different species to compare, and again, there were no significant correlations. And this wasn't terribly surprising. This 40 to 60 micron size class from the images I can see is very diverse and represents many different species. And so for this size class, size is probably not an appropriate proxy for identifying the underlying species dynamics. So in summary, for the total tintinnids, I observed a fairly strong and even spring and fall peaks with a variable smaller winter peak, and that individual tintinnids contributed differently to the overall seasonality. And that when comparison to Lavoie, we're seeing that there's relatively steady abundance over the time series. And for certain species in genera, normalized OTU counts captured similar patterns as image-based morphology, and but size as a proxy for community dynamics needs to be carefully considered. So for my third group that I looked at was the Ciliate mesodinium, which is a mixotrophic ciliate that's common in mid-Atlantic waters, and in high enough abundances can cause red tides and be a dominant source of chlorophyll. It sequesters chloroplasts from its cryptophyte prey and retain transcriptively active nuclei from the prey. And so essentially this is a phototrophic ciliate. And for the further analyses, I used manually verified images because it doesn't do as well with the automated classification. Again, here's a time series of mesodinium over the 10 years, and I wanted to know how do morph types contribute to the seasonal patterns, and do these patterns vary from year to year? And for mesodinium, there's a very strong and high in abundance fall peak, with a more variable in timing and smaller winter and spring events. And we also see multi-year trends in the annual multipliers decreasing and increasing throughout the time series. But what really jumps out when you're studying mesodinium is how variable in size it is. And so like the tintinnids, I treated mesodinium size classes as two different populations. So I separated those larger than 20 microns and those larger or smaller than 20. And what I see when I do this is that these different size classes contribute differently to the overall seasonal patterns. So the smallest mesodinium are occurring, most dominant in these fall events and are most responsible for them. And for the larger mesodinium, while they do occur in the fall, they're responsible for these winter and spring peaks. And interestingly, even though they have differing seasonality, we see similar multi-year trends between the two. So, one historical hypothesis for why mesodinium exhibited different cell sizes had to do with nutrients. And so as inorganic nutrients increase, the cell grows. And in a nutrient-limiting environment, the cell rapidly divides and results in smaller mesodinium. And to look at this, here I have a time series of percent large mesodinium and its contribution to the overall population. And when I overlaid our nitrate and nitrite measurements from MVCO, I saw that yes, there were elevations during some of the winters, but this wasn't consistent over the time series. So I thought that there was little support for the nutrient regulation of size. I then overlaid the temperature cycle, and I saw that at about 10 degrees there seemed to be a switch in assemblage from predominantly small to predominantly large cells, and which correlated with the interannual temperature cycle. And I'll point you to a couple years. So in 2007, it took a while to get cold, and it only stayed cold for a short amount of time. And as you can see, the duration of large mesodinium was also relatively short. And in contrast, in 2008, when it was cold for a longer period of time, we see a longer duration of large mesodinium. And then I'll point you to our warmest winter on record in 2012, and we have one of our shortest periods in time where large mesodinium predominate. And so this led to suggestions of temperature regulation of size. I thought of a kind of a temperature hypothesis where these colder environments are selecting for larger, slower growing organisms that aren't dividing as rapidly. And here's just the annual cycle of the percent contribution of the different size classes. You can see the large Mesodynium here in blue, and the small Mesodynium in gray, which you see increase in proportion with temperature. But recently, Mesodynium has been described as part of a species complex. And the different subclades within the species complex have been associated with different cell sizes. So I then questioned whether there were other underlying causes of seasonal variation with size structure, particularly genetically. Are these colder environments selecting for certain genetic strains? And so I was interested in the Mesodynium major rubrum complex, which is comprised of 8 different subclades. And again, as I said, these subclades have been associated with different cell sizes. And in conjunction with the Johnson lab, we used primers designed to specifically amplify parts of the Mesodynium DNA, which are specific enough to identify these separate variants. And so I then started to compare size. So over here, oh, I'm sorry, I didn't mention, I used 8 different samples, which were then cloned and sequenced to identify the different variants within these samples. So I first compared, here are for the 8 samples, this is a box plot of Mesodynium sizes from IFCB images. Here are the temperature for each of those separate samples. And here's the percent contribution of the different variants to the total clone library. And I'll point you to a couple different interesting stories. In the dark blue here, this is Mesodynium variant A. And in the 3 samples where it dominates, it was 3 springs for this time series. And every time it was associated with cold temperatures, so less than 10 degrees, and larger than 20 micron morphotypes. And this variant was first described around McMurdo Sound of Antarctica, and so suggesting that this variant is particularly associated with this colder temperature niche. And then secondly, I'll point to Mesodynium major, which are the red bars here. And for this 3 samples where it represents around greater than 50% of the total clone library, I saw contradictory evidence. And in one of the winters, when it dominated, it was associated with cold temperatures and larger morphotypes. But then there were 2 falls that I noticed it was associated with warmer temperatures and smaller morphotypes. And historically, this Mesodynium major has been associated with some of the largest cell sizes of Mesodynium. But when it was first described, it had been collected from colder waters around Denmark. And so my research here suggests that maybe there's a possible physiological plasticity with temperature, and so more studies of this cell and cell size with warmer temperatures are needed. And lastly, I saw for 2 samples, Mesodynium variant E in the blue and F in the green, dominating samples associated with warm waters and smaller cells, and so more samples will be needed to see if this is a consistent trend. And we now have about 10 or so samples being sequenced to add to this data set. So in summary, for Mesodynium, for the total population, there's a very strong fall peak, but that the different size classes contribute differently to this seasonality. And that there are distinctive patterns of interannual variation that are similar for these 2 different size classes. Also, there's the evidence of the interplay of genetic variation and physiological plasticity, and that size also needs to be carefully considered because large and small Mesodynium morphotypes were associated with multiple variants, and that possibly sequencing and clade-specific qPCR is needed to unambiguously track these species over time. So in conclusion, I showed you that ciliates are very diverse at MVCO, and that within this diversity there are notable functionalities. There are also, we saw systematic patterns of annual cycles, and in some cases, even when the seasonality was similar, we saw differing patterns of interannual variability. And also at a higher taxonomic resolution, there are distinct patterns and fine structuring of the niches. I also showed you that when temporal variability was assessed by genotypes and morphotypes, there were significantly, these two methods were significantly correlated for many taxa. And that size, while for some of the size classes were significantly correlated, it still needs to be carefully considered. And then I also saw that the interplay of genetic and physiological factors regulating the size structure and temperature relationships for mesodynium also needs to be studied a bit further. And as I was saying, mesodynium can be a very dominant source of primary production in certain abundances. And so it's important to understand how these cells are reacting over time, particularly during periods of time like the winter when primary production can be low. And so as with any exploratory and observational research, a lot of hypotheses and new questions arise. And my work has shown that there are certain aspects of seasonality that maybe in patterns that we may not fully understand. And so a lot more experimental work needs to be done to investigate this. I'd also like to incorporate size and biomass into the time series model. I'd love to get my hands on some mesodynium major and look at its responses physiologically with different temperatures. And that single cell sequencing in combination with morphology will help us to understand the identity of some of the ciliates that we cannot resolve with our images. And right now Emily Peacock has been picking some cells, so we plan on doing this. And another thing I put on here is I'd love to investigate some of the top-down control over time, which I mentioned some bottom-up. And so now I'd like to say some thank yous. Our funding sources from the Gordon and Betty Moore Foundation, NSF, at Woods Hole, the Innovative Technology Fund, and I got some OVF funding, as well as MIT for their support with my schooling, as well as travel funds. First of all, I'd like to thank my advisor Heidi for being so patient and kind and understanding throughout this experience, which I couldn't have done without her. And she's been a great advisor, and not just an academic way, from letting me explore my questions and helping me with my writing, but also helping me through some personal bumps throughout this time. And also stimulating me in times when, there were, sometimes it got tough. And so, thank you for making me a better scientist, Heidi. I'd also like to thank my committee, Rob. Thank you for taking someone who didn't even know how to use a drill under your wing and teaching me how to see a problem, figure out logically the steps to answer it, and also that it was okay to spill water as long as it was fresh. And Becky, thank you so much again for also taking a student who was not very well-versed in the molecular side and trusting me not to contaminate the stocks. And we had such great conversations about how to work with some of this new data. Matt, thank you so much for some of the great conversations we had about mesodynium, as well as your part in my mesodynium project, and also showing me what a live Lavoia looks like under the scope, which I've been trying to do for many years and had never gotten to. Susanna, thank you so much for all your insight into my data and chapters, as well as providing really good thoughts for publication. And also Penny, again, thank you for your insight, as well as some of your advice on observational ecology helped to boost my confidence and direct some of my questions. I'd like to thank my thesis chair, Sam, for helping to put this together, as well as taking me to the Arctic and showing me the magic of how biooptical science can come together to show some really interesting things. Andy Solo for helping to develop the model and chat stats with me when I didn't always know what I was talking about. Also the members of the Sossack and Olson lab, Alexi, who couldn't be here today, but for him no problem is too large, and he is a great source of help with instrumentation and also for chatting. Also Kristen Hunter-Severa, who came up for this, thank you so much for being there for my sanity check-ins and just all of your insight into the data and help with the model and being such a great friend. For Emily and Taylor, our lab cannot run without you guys, and my work could not have been done without you guys. From help from working on some of the images to instrumentation, thank you guys so much. Joe in the lab, who helped to make our data easy to use and easily accessible, which is a major feat. We have a lot of data. And also Ben Lambert, who's been gone for a long time. I'd like to thank Dave Baudouin for helping with the mesodynium sequencing. My classmates, Megan May and Alexis Fisher, for all of your insight into my talk, as well as just being great friends. The colleagues and crew of all of the ships that I've been able to go out on. Also, the WHOI Academic Programs Office, who we students can't do what we do without you guys. Also, the staff at Coffee O for keeping me caffeinated and energized throughout this whole time. And now I'm going to go through a couple pictures. I'll try to be quick. I know this is kind of long. But I'd really like to thank the members of the WHOI security, especially Norman and Linda. I would come in at the weirdest hours for the longest time and just be out on the dock working with my instrument. And they always just had a smile on their face and made me smile in times when it was rough conditions out there at the dock. And I'm going to thank IFCB 14. It's provided me with a lot of adventures, and I really hope that it treats Emily and Taylor well in the future. And also my second office, the trailer out at the dock that always kept me slightly cool in the summer and slightly warm in the winter, only leaked slightly when it rained. But it was a great place to do some of this instrumentation. Also my classmates and cohort, we had a great time throughout this process, maybe not with P-sets, but definitely with Halloween parties. And Kristen, thank you for catching my bouquet. Also my friends back home, they've been a great source of support, and even though we've been far away, they've always made me feel like we're still close. Some of my family who couldn't be here today, my in-laws, as well as my brother-in-law Tony, who are just wonderful sources of support and humor in my life. Also my dad and my stepmom, who also couldn't be here today, they have provided me with so much love and support that is what a grad student needs during these times. And from teaching me to appreciate the natural environment to teaching me how to dance at my wedding, they've been our wonderful parents. And to my mom and my stepdad who are here today, they have nurtured my love of science in such an amazing way throughout this time. And they've always showed how proud of me they are. They've always supported me in anything that I do. Kevin's never afraid to wear a pink shirt for us. My mom has taught Sarah and I how to be decent human beings. And so then, my sister Sarah, thank you so much for all of your support, from sending me flowers during due dates, to listening to my crazy calls, and moving mountains to make sure everything just works in my life. Also, of course, my animals, for all of the support and cuddles that they provide. So my dog Jaffe, Madeline, who sadly passed away, and my little Banjo. And lastly, but of course not least, my husband Mark, who, from driving me at midnight in the middle of a blizzard down to the dock because I was afraid my instrument was frozen, to making sure I'm well fed, filling my life full of art. You've been the most amazing companion throughout this whole thing. And this all started when we were on Ferry to the Vineyard many years ago, and you asked me what I wanted in life, and here we are. So I'm going to next show a corny picture. It's been a very long road, but you stuck with me, and you went out on a limb with me 6 years ago, and for that I'll be forever grateful. So, any questions?
Thank you, Emily. Just for a little bit of perspective, those of you who might not have a background in sort of classical biological oceanography, this is a really underappreciated area of the science, and we're kind of lucky here at Woods Hole to have such expertise in coastology. That's just not the case in most of the oceanographic field. Like, this is really unique, cutting edge, and important stuff in advancing the science. So, some of us are able to answer certain questions about biological oceanography, but Ciliate's have always been sort of an empty
Black box.
you know. And this is innovative work. So that means that you should have plenty of questions for Emily right now. Who would like to start?